My topic was the potential of treating neurodegenerative diseases through the use of nanoparticles. My name is Justin Abraham. I am a senior at Huntington High School in Maryland, and I think let's go into the introduction. The topic I was trying to investigate is, do nanoparticles have the potential to treat neurodegenerative diseases like Alzheimer's and Parkinson's in ways traditional medicine can't? I focused on Alzheimer's and Parkinson's because those are the two main diseases or neurodegenerative diseases. And so I thought those were the two common, and I think that would be more interesting to talk about, and it could be more relative. And another question I wanted to answer was, what makes nanoparticles so special in treating such complex diseases, and does it have any limitations? I also kind of just talked about this. My objective was to explore the potential of nanomedicine or nanoparticles in addressing the challenges posed by neurodegenerative diseases. And it aims to research how nanoparticles can overcome barriers such as the blood-brain barrier, improve therapeutic outcomes, and reduce side effects compared to other therapeutic and traditional medicines. For my methodology, I used the Science Direct and National Library of Medicine, or NIH. My research paper is a literary review or literature review, and so which I just collected data from other research papers and summarized it and how it relates to nanoparticles bringing an effect on or having therapeutic effect on neurodegenerative diseases. So yeah, background information: Alzheimer's, which most know is a memory loss disease. It causes memory loss, cognitive decline, and behavioral changes. And it is primarily caused by the amyloid beta plaque and tau protein that tangles in the brain. Parkinson's is another disease that affects movement, and that is caused by loss of dopamine. That is, caused by loss of dopamine and can cause tremors, slow movement, and muscle stiffness, and it happens in later in age. And I wanted to make an emphasis on how nanomedicine provides a new approach by delivering drugs directly into the brain and bypassing the barriers like the blood-brain barrier. It is designed to target affected areas such as delivering dopamine in Parkinson's and reducing the amyloid plaques in Alzheimer's. My results were basically from all my research, I was able to get that nanotechnology is able to enable targeted drug delivery directly to disease cells, reducing side effects and increasing therapeutic efficiency. Nanotechnology allows for encapsulation of poor soluble drugs, improving solubility and absorption in the body. My picture down here kind of talks about how the nanoparticles are able to bypass the blood barrier, and it is able to, since its unique properties of being small and efficient, and it also can be designed with specific pHs and specific rings, it could bypass the blood barrier and target effective cells, or affected cells by the tau buildup or dopamine loss. Nanoparticles can deliver therapeutic agents to protect dopaminergic neurons or sustain dopamine release and promote neuron survival. And I want to show that one responsibility of encapsulation of drug guarding against enzymatic degradation, targeting the particular cells of the brain passing through the blood barrier and releasing the drug in a specific pH. The other part is the nano-engineered complex. Nanoparticles possess a site-specific drug delivery mechanism to cross the blood barrier, and which is one of the advantages compared to traditional medicines. I wanted to show in my literary review. So it basically consists of talking about how nanoparticles can treat Alzheimer's, and then the second section talks about Parkinson's, how nanoparticles can treat Parkinson's disease, and it's kind of still experimental data. It's not yet being able to be produced widely or in public. So, I want to emphasize two studies or show two studies that talk about it for Alzheimer's. The first study talks about the types of liposomes and polymeric nanoparticles for crossing the blood-brain barrier and delivering drugs. Yeah, I want to show that the main focus was that these nanoparticles can pass the blood-brain barrier in which traditional medicines can't. That's the main effect or main point in which it can, after passing this barrier, it can release therapeutic agents. We can help treat the disease. It also One study also showed effect on how these nanoparticles can release therapeutic agents, reducing the amyloid beta levels and improving cognition function. Certain experiments on certain models and experiments showed significant results in doing that, but yet has yet to be have a significant experiment or result in public view. Parkinson's disease research, one study talks about delivering therapeutic agents to protect dopaminergic neurons in Parkinson's disease. It's only been used on animal models yet, so it hasn't been able to be used in human labs or labs. Nanoparticles cross the blood barrier and improve motor function by promoting neuron survival. The second source or resource I want to put emphasis on was the Gupta resource, where it provides a sustained dopamine release and improves motor function, and has fewer side effects. So it basically just promotes the neuron or the dopamine neuron, which is quite essential for treating Parkinson's. My conclusion, I wanted to talk about, I want to show my importance and contribution. This paper researches nanoparticles, innovative solutions for delivering therapeutic agents across the blood-brain barrier and targeting specific brain pathogens in the neurodegenerative diseases. Nanoparticles have shown significant promise in drug delivery and gene therapy and treatment for Alzheimer's and Parkinson's disease. There is a limitation on the safety of it. A lot of sources show that if misused, this particle can be toxic to other parts of the body, and so that is why it's many years away from being put out in public and public use, or even in like hospitals, because it has to be used in a certain way and by certain technicians to be an effective treatment and it's only used for certain situations or certain cases and very like high level cases. Limitation for my research was due to the fact that I could not run any experiment or lab. My research was purely based on peer review of studies and research papers, and it was not, and the topic would be limited based on the results of the sources I use, not personal conducted studies. That's it.
Wonderful, right? You want to stop sharing and share
Okay.
your experience about the program? Thank you.
Okay. I had a really good time and experience with the program. Professor Lalova really helped me. On like our one-to-one meeting, I think that was like probably the best feedback I got. She really helped me focus on areas I really need to improve on, place I need to expand, what I need to cut out in my paper, and it kind of helped me focus on what I should next time, in the future when I write a research paper. She helped me realize what I should focus on and what I shouldn't, and what is essential in including in my paper content wise. And Professor Virgil was really good at helping me kind of organizing my research paper, going by section with methodology, introduction, and literary review, talking about what needs to be cut out, what needs to be put in, and helping me structure it in the way that most people would want to read it and what professors are looking for. So I thought that was really insightful in my experience.
Sounds wonderful. Question for Justin, Dr. Lidonova.
Very good work there. My question is, so in your opinion, which type of nanoparticles would be the most suitable for drug delivery nanocarriers for this type of diseases?
So I would say liposomes and polymeric nanoparticles because those
Mhm.
are able to pass the blood barrier with little side effect or
Mhm.
very little effect, or sorry, very effective to the brain in one sense, but very
Mhm.
little side effects. A lot of studies I was reviewing showed that even though it was effective in treating like Alzheimer's and Parkinson's, it had a lot of side effects and a lot of unknown side effects in the experiment, which caused like some toxicity or some damage in different organs, not in the brain. So I thought the polymeric
Mhm.
nanoparticles and the liposomes would be good in the case that it was able to pass the blood barrier and deliver the therapeutic agents with not as bad side effects compared to other research and studies I've seen.
Thank you. Great answer.
Fantastic. Two questions for Justin.